My name's Phil Richmond, and today I'm going to show you how to use the Variant Designer tool of GeneBreaker. So GeneBreaker, briefly, is a framework which can be used to create synthetic rare genetic disease cases. Now, rare genetic diseases, especially Mendelian rare genetic diseases, are caused by one or both copies of a gene in the genome having a pathogenic variant, meaning that they contain a variant with which disrupts or changes the function of that gene, and that leads to disease phenotypes or symptoms in the patients. Now, we facilitate the design of one or two variants, depending on whether this is a dominant or a recessive inheritance model, which we can further model the inheritance by using pedigree structure and by assigning which individuals inherit these pathogenic variants. We then export into standardized formats such as PED for pedigree or VCF for our variant calls. And lastly, these tools can also be used or these files can be used for downstream tools to test analysis pipelines or train genome analysts. So if you have gotten here from YouTube, briefly the website is genebreaker.cmmt.ubc.ca, and we're going to jump into the design process. So designing a variant starts off with this page where we have a few different options and then a miniature integrative genomics viewer, which right now is just showing the entire human genome. The first thing we have to select is either reference version 38 or reference version 37, also known as HG19 and HG38. We're going to go ahead and select HG38 or GRCH38 as it's the newer and arguably better reference genome. Next, we choose a proband sex, which is important if we are designing a variant in a gene over the X chromosome or the Y chromosome. Then it'll be important how we choose the sex of our proband or of our affected patient. Then we have the gene symbol, and if you need motivation for selecting gene symbols which have Mendelian inheritance patterns, I suggest you go to the OMIM or OMIM website, which you can find on Google. The gene that I'm going to use is CFTR, which is responsible for cystic fibrosis, which is an autosomal recessive rare genetic disorder. Once I have my gene, I'm going to select Fetch Transcripts, and it'll show me a list of the transcripts with which I can design variants over. We use the RefSeq transcripts from the NIH and supported by UCSC Genome Browser. And for CFTR, right now we only maintain the primary transcript, which is NM000492. So when we click that transcript, you may have noticed that we have now zoomed in our genomic viewer to the entire length of the gene, and this is going to be important when it comes to navigating and designing variants which may disrupt portions of this gene. We can use this by clicking and dragging to zoom in, like I just did there. You can also do it again or use the zoom tool here. And if you continue zooming, you can get all the way down to the nucleotide level, which is going to be important when we design variants, both large and small. So by clicking next, we now are in the variant 1 design module. So we want to select the region of the gene. So genic would be anywhere in the gene, coding anywhere in the protein coding regions, within the untranslated regions or the UTRs, within any of the introns, or within a custom region, perhaps a mix of multiple regions or outside of the gene. We're going to start with looking at an intronic variant, as these have become more appreciated with whole genome sequencing. Next, the type of variant. Well, we can select from ClinVar and ClinGen, which maintain known pathogenic variants, or we can design our own variant from one of these other options, including copy number variant, mobile element insertion, insertion and deletion, at least small, less than 100 base pairs, single nucleotide variant, being just a single nucleotide changing, or a short tandem repeat expansion, where we have predefined the set of repeats which you can choose from, including a set of known pathogenic events. Now I'm going to go ahead and start by using ClinVar. We want to insert a known pathogenic intronic variant as the first variant within this design. So I can choose heterozygous or homozygous. This is a recessive trait, so if I was only designing one variant and I wanted it to affect both alleles, I would need it to be homozygous. But as it's just affecting one allele, I'm going to use heterozygous, and I will design a second variant to disrupt the other allele. Next, I'll click Fetch ClinVar Variants, and a little message will pop up showing that it is looking for them. And now it has found the intronic variants, which have listed their ClinVar ID. They are sorted by start position. It has the reference nucleotide, the alternate nucleotide, and the clinical significance from ClinVar. Now, we can always get additional information about a given variant by going to ClinVar and searching for this ClinVar ID, but I'm going to go ahead and use this ClinVar variant, which is in an intronic region. So I can go ahead and just zoom right into where it's going to be, 117.509.032. And we can see that this variant is right here in the intron, near the border of this intron exon junction. Likely this variant results into the disruption of splicing for this gene. So I think that's a pretty good one to choose. Now the second variant we're going to design, we need to design one, yes, this is a recessive inheritance model. We're going to go ahead and look at creating a variant which will be disrupting the coding region. Now we can design our own variant, which could be an indel, and when you're choosing an indel or an insertion and deletion, we want it heterozygous. Here you're going to choose the start position, so we could start somewhere in this exon right here. Choose one of these start positions, for example 117.509.085, and let's make this result in a frame shift, so it cannot be a multiple of 3. So let's go ahead and make a 4 base pair insertion, or with the negative, this will be a 4 base pair deletion. So if you want an insertion, you're going to add positive, deletion, negative. The other types of variants we can insert include mobile element insertions. So here we could also put a mobile element such as a element type being an ALU, a line, or an SVA as 3 active transposable elements in the human genome. We can go ahead and put a line 1 element right into the coding region of this gene, which likely would also disrupt it. Another thing we can do is we can design copy number variants. So for a copy number variant, if we wanted to zoom out, perhaps we wanted to design our own copy number variant which took out the entire length of the gene. We could give a start position to the left, 117.450.000, and then finish it on the right, 117.700.000. And let's go ahead and delete the entire copy of the gene. Now, if we had done this, then our resulting variant would be on one allele an intronic disruption, and on the other a full-length deletion of the gene. There are some other options which you can choose, and I encourage you to explore the different ones. But for now, we're just going to go with this variant. And the last page that we have shows us a summary of the 2 variants we've designed, and just part of the VCF file, including the chromosome, position, ref, and Alt, more information will be in the actual files. And then the ability to add additional family members. So if the patient today shows up to the clinic and also has a mother, a father, and an unaffected brother, we can then assign each of these genomic variants. Let's assume that the proband inherited variant 1 from the mother, inherited variant 2 from the father, and the brother also has variant 2. Now we're going to download our outputs, including our VCF and our PED or pedigree file. And we are just going to look at those really quick. So if we have a look, we have our 2 variants files, or our 2 files, the first one being the variant file or the VCF file, which has the information for both of these variants that we've designed. And the second will be a PED file or a pedigree file, which has information about which patient is affected, their identifier, their paternal and maternal identifiers, the sex, and the phenotype, meaning that they are affected or unaffected. And with that, if you'd like to start over or create your own case, I recommend trying it. And if you find any bugs, please report it to our GitHub, which you can find on the More Info tab as an issue. Thank you, and goodbye.